Hello, this is the second prep for lecture 22. And I left you with this set of data here. And we had plated them on minimal media that had been supplemented with thymine and purine. So these are the 4 different genotypes that can grow on that kind of media. And we have lots of these. These result from 2 crossovers, and these crossovers those are our 3 genes. The 2 crossovers are both outside of the region we're actually mapping. And because the chromosome is, and the DNA is quite large, there's lots of places where these crossovers could take place. So this is kind of like the non-crossover class for us, because the genes are all exactly the same as the HFR strain, right? So there hasn't been any crossover between the genes. Then we have these 2 categories. These would be equivalent to the single crossover classes if we were doing a 3-point test cross. And so they also come from 2 crossovers, but one of the crossovers is before methionine, and then the second one is somewhere between the genes in question. So either between methionine and thymine, or thymine and purine, or purine and methionine, depending on the gene order here. And then this is our 4 crossover class, which is the same as our double crossover. So we've had crossover between methionine and elsewhere, and then the others, suppose that this is the order here, but we've also had crossovers in between too. So there have been 2 crossovers in the area we're interested in, and that's why it's equivalent to the double crossover class. Okay, so hopefully that's clear. Now we can use this information to figure out the order of the genes, because hopefully you remember that the one that is different from the parental type is the one that's actually in the middle, right? So that means purine must be between methionine and thymine, because we had it wild type on the HFR and mutant on the F-. And so, you've had a crossover to incorporate the methionine gene onto the F- chromosome, but then you have to cross back on here because we've got the mutant form of the purine, but then we have to cross again because we've got the wild type of the thymine, and then our final crossover completes the circle so that our DNA is not degraded. So this is a 4 crossover class, but there's been 2 crossovers in the region of interest, and we've had to have that, and the one that switches place is the one that's between the other 2. Okay, and hopefully that's clear, because that is what we talked about with 3 gene crosses. So I'm trying to really focus a lot on material you've already learned to try to connect you into it. I think it will help reinforce both 3-point crosses and this kind of mapping. So by thinking of it that way, it should reinforce both the understanding of both topics. Okay, so basically we've already determined by what I just did that it seems like purine must be in the middle, because it's the one that's different between the HFR and then the 4 crossover or the double crossover class. But if you wanted to convince yourself, you would draw it out, right? So to confirm it, the order could have thymine in the middle, or it could have purine in the middle. And if we were looking for methionine wild type and thymine wild type but purine mutant, we would get that with just 2 crossovers, right? So we can do that with 2 crossovers, so we'd expect there'd be more of these. And if this was the order, then what would the 4 crossover class look like? It would have methionine wild type, thymine mutant, purine wild type, and then 1 more crossover to complete the circle. So it'd be wild type for these 2 and then mutant for this one. And both of these show that that isn't the correct order, But both of these, I'll fix this. I see it's out of alignment somehow when I full screen it. This one actually does give us, if we do 4 crossovers, then we get methionine wild type, purine mutant, thymine wild type, right? So that agrees. And if we do the 2 crossovers, we get methionine with purine wild type and thymine minus. So for both of these, they all confirm that purine must be in the middle, right? This is one of our single crossover classes, and this is definitely our double crossover class. Okay, so that's the correct order. Now, there's nothing wrong with, and if you're on an exam, oh my goodness, it'll take a lot. Okay, if you're on an exam and you just explain that purine is different from the other genes that the HFR had, that will be good enough. But if you want to just quickly convince yourself of it, or if you're asked for full reasoning, then showing this and showing how this gives you the correct genotype will be good enough. Okay, so I strongly recommend always when you're mapping genes to rewrite in order, because it's just so much easier for you to see where the crossovers are taking place, right? So for this genotype, the crossovers are outside, right, of our region that we're trying to map. So in a way, they don't count anything towards the crossovers. This is like our non-crossover class. And then for this genotype, methionine, and then purine, and then thymine. So that means there's a crossover in region 3. That's of interest. It's in bold because that's the one that's really relevant. And then for this one, methionine, but mutant for purine and thymine. So that crossover would be here and in region 1. But that's the bolded one is the one that's of interest. And then for our double crossover category, a crossover here, and then here and here to give us methionine plus, purine minus, thymine plus, right? And so that would be crossovers in all the regions. The ones in bold are the ones that actually matter for calculations. Now notice this is a 0. So if you forgot to add the double crossover categories, you would still get the correct answers. But I will remind you that will not be the case in something you see on an exam, because I will be testing whether you know whether you redraw the genes in the correct order, and whether you correctly include double crossovers. That's one of the key things. So just think what would happen. Suppose there were 2 of these or something. Just go through and do the calculations of map distances with that information, so that you can convince yourself that you actually, that you know how to properly include those in your calculations. So this is how we do it. The recombination frequencies, we do exactly the way we do every other kind of mapping, which is we just count up the number of crossovers and then divide by the total. And that is the general method for doing it. And so when you've done this work, you know where you've got a crossover between purine and thymine, where you've got the crossover between methionine and purine, and then where these 2 crossovers are. And so you basically just add them up the same way as was done with the 3-point crosses. So in the case of this category, the purine to thymine distance is very small, because there were just 6 out of 338 .018 or 1.8 map units. And so that's shown here. And I actually like to put like a little double-headed arrow or something like a bracket at the bottom when I'm showing a map distance, just because I think it's, for me it feels complete to do it that way, to make a complete and nice map. And then for the methionine to purine distance, it's 52, but notice plus 0, so don't forget that you want to include those. If there were 2 of those, you'd add the 2 here, and that gives you 0.154 or 15.4. map units. So clearly this is not, the way it's been drawn here is definitely not to scale. The methionine to purine distance is considerably larger, about 13 times larger than the purine to thymine distance. Okay, and so you would do your calculations and you could explain them, and then you would draw a map like this. And a map is generally a line. There are little ticks where the genes are located and the map distances are indicated. The double arrows are helpful for that, to just show the particular distance and so on. Okay, so this is the overall procedure then. We obtain a rough map of those genes. We choose a gene that's transferred later than the ones you're trying to map, so this provides our selection. And we just remember we have to use the right kind of media for selecting the transfer of that marker, but then we have to replica plate to test for other markers. We've had some practice on that in a previous lecture, so you understand what kind of media and how we figure out all the genotypes and so on. And then the simpler part is just simply determining the gene order with the data and then calculating recombination frequencies, and of course you're usually asked to draw a map. And we want it to be neat, okay? Now I shouldn't be saying that while I'm using my non-dominant hand and my mouse to draw a map. But we want a fairly nice straight line. We want little ticks. We want to make it really clear exactly where the genes are. If you've got it sort of, something written over here, like, I don't know, do you mean this or do you mean over here somewhere? So just make sure you have nice, clear letters. And then make sure you indicate, okay, this is 2.6 map units. And this is, 4.1 map, or whatever, whatever the distances are. So that's a nicely drawn, except for this, of course, a nicely drawn map with the complete information. Yeah, and one thing I've been trying to look up this afternoon was just, in general, in a lot of eukaryotes, it is thought that a map unit is around 150,000, 150,000 base pairs, although I'm starting to see some different estimates that are higher than that, which is quite interesting. But I can't find any information about the approximate distance, like, I don't think a map unit in bacteria is equivalent in terms of the physical length to the map units in eukaryotes, and I suspect they are smaller. But just for your interest, I thought some people might be interested in knowing that. Okay. So making these HFR cells is when an F plasmid that has an insertion sequence finds another insertion sequence. There's a whole bunch of these around the bacterial chromosome. And then there's this crossover between two circles that just joins them together. And now you've got a great big circle with the little F plasmid inserted in it. And so, this happens with a small frequency. And we have a number of strains that have this. There is a even smaller frequency of this process reversing itself. So the plasmid actually excising back out. So this and this sequence, they're similar, they're the same as each other. And so crossover can happen between them and the little circle can be popped back out. That doesn't happen very often, but sometimes when it does, it actually doesn't come out clean and tidy, but it actually brings a little bit of bacterial DNA with it. And then, so now what you have is we call it an F prime. And we call it F prime because it's not the same as F plus. The prime is talking about extra. There are bacterial chromosome genes on here, and they can now be transferred along with the plasmid into a recipient cell. And what that means is that the genes on the plasmid, and there will be corresponding genes somewhere on the bacterial chromosome. This is kind of like being a diploid. We call them partial diploids because if this was, for example, the genes for lactose utilization, and so are these because the recipient cell is going to have those genes. And if this and this are different genotypes, we can understand something about dominance and recessiveness. We can understand about how the genes actually interact with each other and so on. And being able to make these, or not make these, but isolate them when they occur, is one of the key tools that we use to understand how the lac operon works and the interactions between genes of other systems as well. But we're going to talk about the lac operon next in the class, and so this is a good way to introduce how this happens. Normally, in an organism where there's only one copy of every gene, whether something is dominant or recessive is kind of irrelevant, and you don't know how that would work necessarily. This gives you the opportunity to determine that, as well as some other things that we'll talk about when we talk about the operon. And this just shows you how a lac plus F prime can be made. If there is an integration factor, an IS factor that's close to the lac genes, and if it inserts, and then when it excises the wrong, this piece and this piece are supposed to line up, but this piece has the same sequence. And so if these two line up and they cross over and then generate a circle, again, that circle has picked up some of the bacterial genes, and when it gets transferred to another cell, it's going to transfer those genes along with. And so that is where we'll start the next set of lectures. I am going to stop here because I think, yeah, this is just what I've just been saying. I will just talk about these plasmids in the lecture. I don't think it's necessary to do a prep recording of it because it's a relatively short topic and it's more for your interest. We're all much more concerned about multi-antibiotic resistance than we used to. And one of the reasons why this is developed, first of all, the strong selection because of widespread use of antibiotics for a lot of reasons, some of which aren't even to kill bacteria necessarily. But also the fact that if you look at the plasmids of bacteria that are resistant, the genes have been picked up from lots of different types of bacteria. Because remember, bacterial cells can sometimes just bring up DNA out of the environment and so on. And so I will just go through really quickly in the lecture just a small amount of information about that topic. Some of you are really interested in medical things, and I thought this would be of interest to you. Okay, I'm going to stop.